At ISCT, we were able to present our phase 2 data using ProTrans, our Wharton's jelly-MSC cell product, in 15 patients that we treated in a double-blinded, randomized, placebo-controlled trial in Sweden at a single site. And what we were able to show was that with one dose of the MSCs, that was 200 million cells given to the patients on a single peripheral infusion, that we were able over the year course of the study to maintain beta cell function in these patients, as was seen with delta change in C-peptide area under the curve as our primary endpoint. This was compared to the placebo controls, which demonstrated a kind of a downward trend, as would be seen with progression of the disease over a year period. The patient cohort that we utilized was 18 to 40-year-old adult patients, and they were early diagnosed patients, so within 2 years of diagnosis. Moving forward, we also saw encouraging data in our secondary endpoints that indicated that in some of the patients, we actually saw an upward trend in beta cell function. And therefore, we are now moving towards a phase 3 trial where we will evaluate these factors in a larger patient cohort over multiple centers across Europe and evaluate whether we are able to maintain beta cell function in this cohort and move also towards a pediatric population as well, with a currently approved trial that we have starting in the fall. And then we will move towards hopefully market authorization of our product inside Europe.